So today we are going to talk about genetic aspects of health and disease and immunity, and if the time permits, we will also talk briefly about the memory. In a family, every individual has same eye, hair and skin color. This will be similar to his or her parents. The question is why? Because these characters are passed from one generation to other. The same is with diseases. Some of them are passed down from one generation to the next generation. In many cases, diseases or related problems do not have one single cause. It may have a combination of genes as well as their environment. Normally, most genes are copies of the genes present in our parents and work in a similar way as they do in the parents. But sometimes a gene which goes from parent to children is not a perfect copy, means there is some change in their structure. These changes are basically called mutations. Some mutations are better or they do not affect any ill, but some may cause problems or may cause disease. These mutational changes in one gene may develop a genetic disorder, normally called single gene disorder, but if more genes are involved, then it is known as a multiple gene disorder. But some most common diseases are caused by changes in a combination of genes. For example, there is a disease which is caused by a rare a group of rare genetic disease caused by mutations in one gene is known as a cystic fibrosis or CFTR. Mutations can happen anytime during a person's lifetime, and these mutations are always called acquired. Acquired mutation can be caused by environmental factors such as ultraviolet radiation which is received with the sunlight, drugs, exposure to X-rays for a long time, or exposure to high radiations. The acquired mutations are thus different than heritable mutations. They may cause problems like skin cancer, but one cannot necessarily pass them to their children, and that's why they are called as acquired. Basic question emerges here is that how do mutations cause disease? In this diagram, you can see the mutations are of three types. Substitution means a new group is added, and by the addition of this group, the new amino acid added in the protein result into the change of a structure of a protein and function of the protein. Similarly, insertion, a large group of nucleotides are added into a DNA, like similar nucleotides or a different combination of nucleotides, changing the protein sequence again, or there may be a deletion or removal. There is also a possibility of alteration, like a sequence present at one place will move to another place. So these kind of mutations are basically responsible for the abnormal functioning of a gene and known as mutations. Genes usually code for proteins, I already told you, and these molecules perform wide variety of specialized tasks in the cells as enzymes, hormones, structure proteins, etc. These alterations in genes are responsible for more than 5,000 clearly hereditary diseases. To name few: hypertrophic cardiomyopathy, sickle cell disease, cystic fibrosis, Huntington's disease, Parkinson's disease, and also the cancer. Similarly, altered sequence of many other genes influence the development of common illness that arise through the interaction of genes with the environment. Thus, we can show that the environment can also directly cause changes to DNA inside the cell. Where here is an example. Exposure to the sun damages DNA in the cells, and if this damage goes unrepaired, gene changes will be copied into new cells body creates regularly. Though sometimes body repair the gene to help protect itself from developing a disease. Only a small number of mutations cause a genetic disorder. Mutations can sometimes even have a positive effect, but as a resistance against disease, although this is seen rare. We all have two copies of each gene. We get one from each parent, as I already said. In some cases, people are born with one only, or three or more copies of a particular gene. It is also possible for that a particular gene is entirely missing. Absence of a specific protein normally encoded by this gene is directly linked with development of a disease. One of the best example of this is a report from Britain about a boy who had missing gene which was responsible to produce an enzyme called adenine deaminase or ADA, which actually resulted in loss of immunity. This boy was kept in a bubble so that he should not directly come into the contact of environment. This disease was later described as SCID, severe combined immunity syndrome. This is a diagram of the boy who was placed in a bubble, and the doctor has to approach through the small opening inside to talk to the boy or give the medicines to the boy. Sometimes chromosome gets broken, and this broken piece can end up in one cell but missing in the another cell when the two cells separate. This is similar to having an extra or missing copy of gene. These can cause problems related to development, growth, and functions of the body systems. Here is a normal gene producing a normal protein, but if there is a mutated gene, means if there is an alteration, if there is insertion, if there is a deletion, in that case, since the structure of gene is mutated, abnormal protein is produced, and if the gene is absent, no protein is produced. Sometimes what happened, at a single point there is a change in a nucleotide sequence. Only one nucleotide is changed, and this kind of situation is known as a point mutation. In the second panel, we have shown the breakage of a chromosome. You can see very clearly in the middle section. Okay, thank you very much. Now we are going to talk about genes and impact of immunity. What is immunity and why do we require immunity? Well, the word immunity derives from a word called immunis, which is a Latin word, and meaning exemption from military service, tax payments, or other public services. It is a complex biological system that can recognize and tolerate whatever belongs to the self and to reject what is foreign. Well, question here is what is self and what is non-self? See, in our entire body, we have a large number of different types of cells. Each cell is made up of different kinds of proteins, carbohydrate, lipids, enzymes, hormones, and other things. If anything enters into the body, and if the body cells consider it as coming from outside, and the structure is not related to anything like a protein, like a carbohydrate, like a lipid present in our body, in that case, the body recognizes it as a foreign, and therefore it is also labeled as non-self. But if something happens, even the bacteria, even the virus, if it enters into the body, and body recognizes it as a self or as its own structure, in that case, body will not react against it, and that substance will be called as self. In earlier lectures, I also referred that in our intestine there is a bacteria called E. coli or Escherichia coli. This small bacteria is present for a long time in our intestine. Do you know that the Escherichia coli also produce a toxin? But still it is present in our body right from our birth. When you take antibiotics, the amount of bacteria get reduced, and then you have a disturbed digestion or disturbed other activities in the intestine for some time. So this bacteria, which is residing in our intestine for a long time, is known as a self. So in this way, the body differentiates between a self and a non-self substance. Non-self substance can be microorganism, as I said, bacteria or viruses, or to any chemical we talk about, or any drug which we talk about. Some drugs are good, but some drugs may create problems. Even a small piece of a cotton, a small thorn which is entering into your body, sometimes a poison, and different type of foods which can affect your body. They are all labeled as non-self if they are able to provoke any kind of reaction, ill reaction inside the body. Body identifies non-self as a foreign and always fight against it to neutralize its effects. If body identified as similar to body component means self, then it will accept it and will not react against it. This entire system which is present in our body, recognizing the foreign harmful substances and the self or the cell substances is basically called as a defense mechanism of body. It is exactly a system like other systems in our body: circulatory system, excretory system, nervous system, or any other system, muscular system, or excretory system, or reproductive system. So the way the system works, the body's defense system or system also works in the similar fashion. And that's why another name is given to it, is known as the immune system. This has both components, specific and non-specific components. Let us talk about the non-specific component first. The non-specific component always act as a barriers. It eliminates wide range of substances, including microorganisms, which could be pathogens, irrespective of their makeup, several types of drugs, several types of chemicals, and all other substances which I mentioned as non-cell substances. How this substance acts as a barrier? Let us talk about it. Well, actually this is the first step to act against the non-self by the body. Here I just want to add something. You have seen Mahabharata, the picture on the TV, and you have seen the fight between the Kaurava and Pandavas. And then you might have seen that there are the army personals which are on the foot fighting against each other. Then there are horses, then there are elephants, and then there are the firearms and other things. So actually the fighting is like a slowly fighting is like this only, and the immune system also works in a similar way. First there are cells which fight against the substance which enters into the body. If cells are not able to destroy them, then there is something else, and if still it is not possible to destroy them, then something else, and finally if the non-specific substance or non-cell substance is able to escape, in that case the disease will develop, or something illness will develop, or pathology will develop in our body. Every substance, especially the chemical substance, have some chemical groups by which they are recognized by these cells. These are always protein groups, and they are called epitopes. The chemical structure on which the epitopes are present are basically called as antigens. The antigen word is always used especially for the protein substances. Initially, neutrophils, monocytes, etc. identify the epitope and react against the antigenic structure. Well, as in the first defense, the cells are involved. This type of immunity is known as cellular or cell-mediated immunity. But if specialized cells like WBC, which are known as white blood cells in our body, also known as, and when they are modified in the body, when the cells come in contact with the non-cell substance or antigen, they are converted into plasma cells or modified into plasma cells. And these plasma cells are actually, when identified the epitopes, they produce a glycoprotein substance known as antibodies. If the antibodies are produced in the blood which fight against the antigen or the non-cell substance, then such reaction is called as humoral antibody. So basically in immune system, there are two types of reactions. One is cellular cell-mediated immunity reactions, and there is humoral immunity reactions. Reaction or binding of antigen and antibodies is always very specific. Why? Because antibodies are always produced against specific antigen. They are not normally produced. They are produced, but the antibody which is fighting against the antigen are specific antibodies, or they are produced against that particular antibody by recognizing the structure or epitopes present on that by the plasma cells. Reaction through antibodies is called specific immune reaction. The immune system also has two components. One is known as innate and another is known as adaptive. Innate is present in all metazoans, means all multicellular systems. And this innate system always remain in them right from the birth. Small things, I will give you an example. Presence of oil glands with our hairs, maybe on the skin, maybe on the head. All hairs have oil glands and they secrete oil. Hairs present on the skin, production of sweat, saliva which is secreted in mouth has a alkaline nature. Acid present in a stomach is a very strong acid which is HCl. Even if you dip your finger in it, the finger will burn because the pH of this acid is 1.0. Similarly, secretion in ears and glands present in the eyes known as the lacrimal glands also secrete liquid. All these substances or structures present in our body actually help as a first defense against the non-cell substance trying to enter into body. In addition to that, when the non-cell substance try to enter into the body, there are mast cells in the blood. They immediately get burst and there is a substance histamine which is released, and therefore there is an inflammatory reaction happens, so that the site at where this insect has bite or the non-cell substances try to enter into it, or the pathogens try to enter into our body, the redness develop and there is always a itching pain. These are some of the examples of innate immunity. And as I said, they all help as a first line of defense in our body. Innate immunity is also known as native immunity. In addition to innate immunity, additive immunity only occurs in vertebrates. It is more advanced and involves lymphatic cells. These are white blood cells, monocytes, they are largest cells in the blood in comparison to the WBC and the RBC. Neutrophils, also large cells, basophils, acidophils, etc. If you prepare a thin film of a blood on a glass slide and observe under a microscope, you can recognize all these cells by their specific structures. These all cells are able to recognize non-cell substancing coming into our body. Adaptive immunity is also called as acquired immunity. The reason is very simple. It is acquired because this immunity develops due to experience. Means innate is present in our body right from the birth. But this type of immunity which is developing in our body is because of an experience. Means suppose if we have some insect biting at our skin, the cells present in the body will try to recognize the poison which is injected by the insect. And if by action of a cell or action of antibody, the reaction is subside or neutralized, but when after some time or after a long time, the same insect will bite and inject the poison into our body, the reaction, the body will immediately react against it. The reason is very simple, because the cells present in the body have already recognized the poison, structure of a poison or any invading substance, and therefore this time the reaction is more quick. Suppose first time it might have taken about 30 seconds, this time the reaction will be immediately, like in 5 seconds, and it will be more intense as compared to the first time. The reason is very simple, the body has already recognized the structure. This type of phenomena, or remembering the antigenic study, is also called as antigenic memory. Unlike the innate immunity, the acquired immunity is highly specific to a particular pathogen, because it always acts against a specific type of antigens or non-cell substances. The acquired system includes both humoral immunity components as well as cell-mediated immunity components, because initially the cell-mediated reaction takes place and then the humoral immunity component takes place. Adaptive immunity can be acquired either naturally, that is by infection, or artificially through deliberate actions such as vaccination. Therefore, it is actually divided into two groups. One is known as active or passive. Here in the diagram I have exactly explain that how the immunity is classified. Immunity is two types: innate immunity and adaptive immunity. Adaptive immunity is further classified as a natural and artificial. Natural immunity is again identified as a passive and active, and artificial as a passive and active. Well, here I have written in first case natural, passive and maternal. We have to understand this. Maternal means when the child is in the womb of a mother. In that case, it is connected with the mother. So it is receiving the feed through the blood of the mother, which is reaching to the child. So along with this blood, the antibodies which are present in mother, especially IgA, reach to this child. So in initial phase, the child doesn't have a specific immunity inside him or her, and therefore these IgA antibodies which are receiving through the placenta and coming to the child are basically helping the child to protect itself. That's why it is known as the natural. The next immunity is active immunity. Means sometimes you get an infection, a viral infection or a bacterial infection, and therefore the body gets react against it, and therefore the antibodies are produced inside the body and they will remain in the blood. So next time, as I said, if they come, the body will react against it. So by insect bite or by through a water or through a food or through some other source, if you get in touch with a pathogen, this type of immunity is known as natural active immunity. Artificial immunity, passive antibody transfer. How it happens? See, if suppose person has a weak immunity, if we transfer a blood from one person to another person, or we take out a blood and concentrate it, it consists of large number of antibodies, and if we transfer this antibody to another person through blood transfusion, this will be a passive transfer, known as artificial passive transfer. And the another is active immunization, means using a vaccine. By injecting a vaccine, you are get immunized. As similar in the case of a polio drop is given, which is basically consists of polio virus. This polio virus is actually made inactive by either treating with a very high temperature or by keeping at a very cold temperature. So the virus becomes completely inactive, but its DNA remain active. Okay. So in one way, this is live, but it's non-functional totally. So when this polio virus is actually in the form of a vaccine injected in your body, the body will try to recognize its DNA or this virus coat outside where epitopes are present and synthesize the antibody. These antibodies remains in your blood, and when the polio virus attack you, these antibodies will act against it. So this is known as artificial active immunity. When our immune system doesn't work the way it should normally, condition develop may be called as immune system disorder. They may be of several types. Primary immune deficiency, when born with weak immune system. Acquired immune deficiency, when a disease caused basically weakens the immune system in our body, as you can see in many cases. Allergic reactions, when immune system is too much active, and it get active when it comes in contact with a pollen or a dust or a mold, something other things. Autoimmunity, this is very important. When immune elements such as certain specific cells or antibody start, which is present in our own body, start acting against body systems. This type of immunity is known as autoimmunity. Some common examples of immune system disorders are as follows, which provide an insight as how genetics is related to immunity. These are SCID, called as severe combined immunodeficiency syndrome. I already given the example. AIDS, you already know about it. Acquired immunodeficiency syndrome, which is caused by the virus HIV. So human immunodeficiency virus. Asthma, which is basically due to the overactive immune system. Type 1 diabetes, and then ultimately the arthritis. These are the basically diseases which are related with the immunity. Severe combined immunodeficiency. Very briefly, let us talk about these few diseases. Severe combined immunodeficiency disease, also known as SID, is an immune deficiency that is present at birth, and children with such deficiency are in constant danger of infections from bacteria. And I give the example of a boy, Bob, which was kept in a sterile environment inside a plastic bubble. Temporary acquired immune deficiencies, also known as a TAID, and this is basically immune system can be weakened by certain medicines or treatments. The cancer medicines which are given to the patient always reduce or weakens the immune system, and therefore the immune activities are given from outside along with the medicines so that your immune system remain constant. Human immunodeficiency virus which cause the disease is an acquired viral infection that destroys important white blood cells and therefore weakens the immune system. People on as I already told you that people on chemotherapy or other drugs used to treat cancer may also have temporary or low immunity. People with AIDS or some other diseases become seriously ill with infections that most people can just simply fight off. These infections are called opportunistic infections because they take advantage of our weak immune system. Here is a diagram which shows the different type of cells which are in our body. They are generated from the cells which are present in our bone marrow, or which is present in our all bones, and large number of cells like red blood cells, platelets, myoblasts or monocytes, neutrophils, eosinophils, basophils, and then there is a lymphoblast which is again differentiated into B lymphocytes, T lymphocyte and natural killer cells. All these cells are basically responsible for the cell mediated immunity. But WBC, when converted into another cell known as plasma cell, it is responsible for the humoral immunity. In addition to the immune system which is present here, we also have another system known as a lymphatic system. Lymphatic system present in the form of a tonsils, in the form of adenoids, and in form of a small gland known as thymus. In addition to that, in every part of a body there are lymph nodes. Sometimes you have a swelling where you feel some kind of a itching, and this is because the lymph node get the infection and it becomes swells. Lymph node or lymphatic vessels, they are spread throughout the body, as you can see in this diagram, and play important role in fighting against the pathogens entering into the body. In this panel, I have shown the innate immunity and adaptive immunity, and you can see the large number of cells which are involved in the innate immunity and adaptive immunity, where macrophages, dendritic cells are there, then B cells and T cells, they are basically causing the response. Just to show that here is a large cell which is basically a cancer cell. It has large number of fibrous structures, and then the small gray like colors structures which are basically the cells which are phagocytes in nature. Phagocyte means they can engulf anything which is present inside the cell or enters into the cell, like a bacteria enters into the cell, so these phagocytes can enter into it. In the next panel, you can see I have written here pathogen, which is basically a bacteria, and the phagocyte cell is trying to come in contact with it and ultimately will engulf it. What is allergy? If one is born with certain genes, immune system may react to substances in the environment, and these substances are called allergens. Dust, mold, pollens and foods are all allergens. In addition, smell, color, even a sight of a person or sight of a particular scene can cause allergy. Asthma. This is also an immune disease in which coughing, wheezing and trouble in breathing is observed, and this can be triggered by common allergens like dust or pollens, or by an irritant like tobacco smoke. An allergen causes an itchy rash known as atopic dermatitis, and this is also called eczema. Allergic rhinitis is basically sneezing and running nose and swelling of nasal passages, and it is because of the indoor allergens like dust and the hairs of a pets, or outdoor allergens like pollens or molds. In autoimmune diseases, the body attacks its own normal healthy tissues, as I already said, but why they do so is still unknown. It is probably a combination of person's genes and something in the environment that triggers those genes. Three common autoimmune diseases are type 1 diabetes, the immune system, where the immune system attacks the cells in the pancreas that make insulin. Insulin removes sugar from the blood to use as energy. Rheumatoid arthritis, this is a second autoimmune disease Arthritis causes swelling and deformities of the joints. An autoantibody called rheumatoid factor is present in the blood of some people, which cause rheumatoid arthritis. There is another disease known as lupus erythematosus. This disease attacks body tissues, including the lungs, kidneys, and skin, and many types of autoantibodies are found in the blood of these people. No one knows exactly what causes autoimmune disease, but many factors seem to be involved. Scientists have previously identified genes important in controlling the autoimmune response. Thank you. Okay, since we have time, let us talk very briefly about the memory. It's very interesting that how memory is basically governed by the genes. A question always comes to our mind, how we know things we have never learned. First example, Leslie Lemke was a blind American savant, a learned or talented person in the field of art or science. Notable for his work as musician, even though he has never had a music lesson in his life. His musical genius erupted early and spontaneously when he was an infant. So in other words, we can say that it was factory installed. Professional musicians witnessed and confirmed that Lemke somehow, even in the absence of a formal training, had innate access to what can be called as the rules or vast syntax of the music. This is very surprising, that how Lemke has known all these things without a formal training in music. A second example or classical example of this kind is Alonzo Clemons. He is also an American savant and basically is a sculptor and boulder coming from Colorado, USA. He suffered a severe brain injury as a child, which left him with a developmental disability. After the injury, he began to sculpt with whatever was handy or available to him. Can you believe? Now he is a celebrated sculptor who can mold a perfect specimen of any animal with clay in an hour or less after only a single glance at the animal itself. And you can see every muscle and a tendon of that animal will be perfectly positioned in the sculpture which he will make. Interestingly, again, he has no formal training of sculpturing. These two classical examples clearly explain that the memory is genetic, or there is some involvement of genes in this kind of a memory. Genetic memory is those complex abilities and actual sophisticated knowledge inherited along with other more typical and commonly accepted physical and behavioral characteristics. A well-known scientist, Marshall Nirenberg, from the National Heart Institute, USA, who was also involved in first time reporting the artificial gene along with Dr. Hargand Khurana, provided insight into the actual DNA RNA mechanisms of this innate knowledge in an article published in a very famous journal called as JAMA in 1968, titled Genetic Memory. There are very classical examples for this. In an animal kingdom, there are ample examples of such complex inherited capacities. Monarch butterfly, I think we have seen many times. Very colorful and beautiful butterfly. It's a small butterfly. This butterfly makes a 2,500-mile journey from Canada to Mexico every year, where they stay for winter. In spring, they begin the long journey back north, but it takes 3 generations to do so. Means after 3 generations, they return to their back home. So no butterfly which is making the return journey has flown that entire route before. How do they know a route they have never learned? That means it has to be an GPS-like software inherited from their parents. There is another example of songbirds. These songbirds are known as oscine. These are perching birds. They are also called oscines, and they are found throughout the world. They have vocal organs typically developed in such a way to produce a diverse and elaborated bird song. They have voice box with more complicated parts than other birds. So they can sing especially musical and sometimes beautiful songs. Bird uses these songs to attract mates or to demonstrate territories. Different kind of birds of these species sing different songs. Passerine birds, house sparrow or sparrows, they all come under the songbird category. Songbirds learn singing intricates early in life. So how do young birds learn how to sing? Birds either calls or sing. Calls are always small, but song is for a more time. These characteristics I studied in 1950 showed that nestlings of these birds raised without exposure to adult males develop abnormal song. But when they were exposed to sound recording of adults, birds were or nestlings were able to sing specific song characteristics to their species at the adult age. This demonstrates that birds might have learned songs in early life or might have an innate type of learning of songs. Surprisingly, even if these birds are raised in soundproof isolation, they can give the usual call for their species with no formal learning. One of the trickiest questions in biology could be, can memory be inherited? Well, normally we will say no, it's not possible. But researchers have demonstrated that memories are stored in the brain in the form of a neural connection or synapses, and there is no way to transfer this information to the DNA or a germ cell. I will explain it in brief. It is basically explained that the memory, earlier it was considered or it was reported that the memory, whatever we learn, is actually stored in the neurons cytoplasm in the form of RNA or small proteins. But later it was discovered that the memory is basically in the form of small fiber-like structures which are present on the neuronal fibers. The number of these fibers increase with the increase in learning or increase in memory. But suppose if this is a question, if this is a thing that the memory is stored in the form of a fibers which are present on neuronal connections or synapses, in that case, is it possible to transfer it into the next generation? Well, actually it is not possible. The reason is, if you want to transfer anything from one generation to another, this thing should go to the germ cells. Means the testes should form a gamete and the ova should form a gamete, and when they fuse together, then ultimately the individual is formed. So these are basically the germ cells formed from the testes and from the ova, and there is no way by which these kind of structures, the fibers which are present in neural connections or synapses, can be transferred to the germ cells, or DNA related to this can be transferred to the germ cells. We do not inherit the French or German our parents might have learned at school, because we have to learn it ourselves. But at least in some species, certain forms of memory can be transmitted to next generation. In 1962, biologist James V. McConnell published a controversial experiment in which he claimed to pass on learning process from one worm to another by feeding the later the ground remains of the former worm. And he was convinced that the engram of a memory, which is basically a hypothetical term for the physical trace of memory, resided in RNA molecules, which could transfer that memory from the one worm to the another worm, which we did. Although McConnell's experiment was later completely discredited by scientific community, but shortly thereafter, other studies showed transfer of memories from one rodent to another through injections of brain extracts. In recent decades, it has become known that not only RNA molecules are also involved in formation of long-term memory. In May 2018, a study conducted by David Glanzman, University of California, Los Angeles, showed that injection of an RNA extract from specimens of a sea slug, Aplysia californica, trained to respond to an electrical stimulus, was capable of transferring this learning to other untrained individuals. He also showed that the RNA of the first animal stimulated the neurons of the second animal isolated in a petri dish. Glenchman says that it's as though we transferred the memory. If memories were stored at synapses, there is no way our experiment would have worked. In another experiment, artificial method of straight injection was used to transfer the memory. If we presume RNA as a form of memory to be inherited by children from their parents, to be possible for this, a pathway must first be opened for the transfer of RNA from neurons to germ cells. But this is precisely what a team of researchers at the University of Maryland did in 2017. They discovered that in the free-living worm known as Canor aviatis elegans, certain strands of RNA produced in neurons can travel to germ cells and silence genes in offspring even up to 25 generations later. David Ornstein in July 2021 published that memory making involves extensive DNA breaking. These small DNA breaks, also known as DNA double-strand breaks, because DNA is double-stranded structure and therefore there is a small breaks at various lengths of the DNA. These are called as a DNA double-strand breaks. According to DNA, similarly to Li Hu Tsai, professor of neuroscience at MIT, attempted to understand exactly how widespread and extensive this natural activity in the brain upon memory formation. But findings mostly made in lab preparations of neurons, they did not capture the full extent of the activity in the context of memory formation in a behaving animal and did not investigate what happened in cells other than neurons. A study published in PLOS One by Ryan Scott and Oleg Kirsi also showed that the DSB activity is involved in learning and memory. They gave mice little electrical jabs to the feet to condition a fear memory of electrical jabs, and they then used several methods to assess the double-stranded breaks and gene expression in the DNA in the brains of the mice over the next half hour, particularly among a variety of cell types in the prefrontal cortex and especially hippocampus. Two reasons essential for the formation and storage of conditioned fear memories. They also made measurements in the brains of mice that did not experience the food shock to establish a baseline of activity for comparison. These studies clearly suggest that the double-stranded DNA breaks are also related with the memory formation. The creation of a fear memory doubled the number of DSBs among neurons in the hippocampus and the prefrontal cortex, affecting more than 300 genes in each region. Among 206 affected genes common to both regions, the researchers then looked at what those genes do. Many were associated with the function of the connections neurons make with each other, which are called synapses. This makes sense because learning arises when neurons change their connections. This is a phenomena known as synaptic plasticity, means molding your own brain or molding a brain in the way of we want. And memories are formed when groups of neurons connect together into ensembles called engrams. Thank you.